Hello, my name is Andrew Tapia. I'm a PhD student in the Department of Computer Science at University of Kentucky, and I'm presenting my poster on RNA-clique, a new method for computing genetic distance matrices from RNA-seq data. This method was developed both by me and by my advisors, Drs. Uromchuk and Moore in the Department of Computer Science and Dr. Shardle in the Department of Plant Pathology. Genetic distances measure the degree of difference in the genomes of some set of individuals. Although it's natural to use DNA sequence data for this purpose, RNA-seq data could be an economical alternative since RNA sequences mostly reflect the genomic DNA sequence, and RNA-seq data are commonly obtained for studies of gene expression. The method begins with RNA-seq data for some number of samples, representing, for example, individuals. We then assemble the RNA-seq reads into transcriptomes. For testing, we used RNA spades, but you could potentially use other assemblers. The next step does require that each transcript be associated with some Kamer coverage value, quantifying the amount of input sequence that goes into the assembled transcript. The next step also assumes that the set of transcripts is partitioned into sets of transcripts referred to as genes. Each transcript belonging to a gene is assumed to be an isoform resulting from alternative splicing. The next step is to select the top n genes for each sample by Kamer coverage. Here n is a configurable parameter. We define the Kamer coverage of a gene as the maximum Kamer coverage among the gene's transcript isoforms. Next, we blast the top n genes for each sample against those for every other sample. From this step, we obtain a table of blast hits for every ordered pair of distinct samples. For each unordered pair of samples, we use the pair of blast results for those samples to produce what we call the gene matches table for that pair. The gene matches table identifies pairs of genes for that pair of samples that are likely orthologs. We then use the gene matches tables to build a gene matches graph. Each vertex in the gene matches graph represents a specific gene in a specific sample. An edge exists between two vertices if that pair of genes appears in some gene matches table. The next step of the algorithm classifies the connected components of the graph according to the number of vertices and edges in the components. We define a small component as one with fewer vertices than samples, and we define a large component as one with at least as many vertices as samples. An ideal component is one that has exactly as many vertices as samples and is a clique. That is, it has an edge between every pair of its vertices. These ideal components represent genes that have orthologs in every sample. Using the gene matches graph, RNA-clique filters the gene matches tables to include only rows for gene pairs appearing in some ideal component. Finally, we compute the genetic distance for each pair of samples using the filter gene matches table for those samples. The similarity of two samples is defined as the total number of identical nucleotides for all hits in the table divided by the total lengths of all alignments, excluding gaps. The distance is then defined as 1 minus the similarity. We tested RNA-clique with 3 different data sets. The first data set was a set of 16 samples of the grass tall fescue. Each sample within that data set belonged to one of 4 different genotypes. The second data set was a set of 24 samples of the teleost fish bluehead wrasse. For that data set, we had samples from 12 individuals, and for each individual, we had samples from 2 different tissues, one from the midbrain or forebrain tissue and one from gonad tissue. The third data set was a set of 16 simulated transcriptomes that we generated using the library DendroPy. We first generated a ground truth phylogeny using the birth-death process model, and then we generated 50,000 random transcripts and simulated their evolution along the tree using the HKY85 evolution model. We also tested an alternative method, cnidaria, on the first 2 data sets. We visualized the results for the tall fescue data as PCOA plots. For both RNA-clique and cnidaria, samples clustered according to genotype. Samples within clusters tended to be less spread out for RNA-clique than for cnidaria. Since we expect samples of the same genotype to have near zero distance, this may mean that RNA-clique gives slightly better results than cnidaria for this data set. For the bluehead wrasse dataset, the most similar samples according to RNA-clique were almost always those from the same individual. The exceptions were the samples from individuals 52 and 114, but we believe these exceptions are the result of labels having been swapped in the NCBI sequence read archive. For cnidaria, we see that the distances reflect differences in tissue type. Of course, this is not what we want to see for a genetic distance, so we conclude that RNA-clique also gives more reliable results for the bluehead RAS dataset. For the simulated transcriptomes, we used the genetic distance matrix obtained from RNA-clique to build a phylogenetic tree using the neighbor joining method. The resulting tree was topologically identical to the ground truth. In conclusion, the results show that RNA-clique works well on both of the real datasets we've tested as well as the simulated dataset. Moreover, based on our comparison, we conclude RNA-clique gives more reliable results than the existing method cnidaria. The software implementation of RNA-clique is available for download from the RNA-clique GitHub repository at github.com/actapia/RNA_clique. The GitHub repository provides instructions for installing and using the software, as well as a tutorial explaining how to get from RNA-seq data to PCOA plots, trees, and heat maps. This work was supported by funding from the U.S. National Science Foundation. For further information about RNA-clique, please also see our recent article in BMC Bioinformatics. Thank you.